Good morning to all the participants joining us today. On behalf of Federation of Clinical Pharmacists in India and Clinical Research Forum, I'm delighted to welcome you to today's session of our basic module in clinical research titled Introduction to Clinical Research and Its Importance. Good morning to all the participants joining us today. On behalf of Federation of Clinical Pharmacists in India and Clinical Research Forum, it gives me an immense pleasure to introduce our esteemed speaker for the day, Dr. Haritha S. Nath, PharmD. Dr. Haritha is currently serving as an assistant professor at the prestigious Dr. D.Y. Patil Institute of Pharmaceutical Sciences and Research, Pimpri, Pune, a dedicated academician and clinical researcher. She has been actively involved in pharmacy education and professional development since 2021. She holds a Doctor of Pharmacy degree from NGSM Institute of Pharmaceutical Sciences, NITE, deemed to be University Mangalore, and has contributed to the field through research publication in reputed journals. She has presented her work at numerous national and international platforms and continues to guide students in research. Notably, Dr. Haritha also serves as a faculty advisor of the ISPO DPU student chapter and is an active innovation ambassador under Institution Innovation Council. With her passion for clinical research, patient care, and teaching, she brings a unique blend of academic depth and practical experience to today's session. Let us warmly welcome Dr. Haritha S. Nath as she takes us through the fundamentals and vital role of clinical research in today's healthcare landscape. Over to you, Dr. Haritha. And teaching, she brings a unique blend of academic depth and practical experience to today's session. Let us warmly welcome Dr. Haritha S. Nath as she takes us through the fundamentals and vital role of clinical research in today's healthcare landscape.
Today we
Over
will
to
be
you, Dr.
looking
Haritha.
after the
And teaching, unique blend of academic depth and practical experience to today's session. Let us warmly welcome Dr. Haritha S. Nath as she takes us through the fundamentals and vital role of clinical research in today's healthcare landscape.
Today we Thank you, Sajal. Thank you for the nice introduction about me. So today we will be discussing about the introduction to clinical research and its importance. So first we will see what are our learning objectives of today's. So first one, to understand the concept and purpose of clinical research. So what is mean by clinical research, and in clinical research, what we will be doing? So we will be understanding the concept of clinical research today, and we will be going through the history of clinical research, how it started and how it got evolved. Then we will be discussing about the different types of clinical research as well as phases of clinical studies, phases of clinical trials. Then we will be looking on to the ethical and regulatory framework involved in clinical research. Also, we will be discussing about the importance and future trends in the field. So first, what is clinical research? So clinical research is a component of medical and healthcare intended to produce knowledge valuable for understanding human disease, preventing and treating illness, and promoting health. Basically, if you're doing clinical research, we will be able to understand what is the mechanism of a new entity, a new molecule, a new drug, and how it is helping in like disease prevention or treating the disease and promoting the overall quality of life of a patient. Okay, and clinical research, the major point of clinical research is all the research where the humans are involved, it is called as clinical research. So if the animals are involved, we are having a different name, preclinical research, and in vitro is also there. So in vitro, in vivo, and after that comes the clinical part where the humans are involved. So all the research which is taking part in humans will be accounted for the clinical research. So it may be in a healthy volunteer or as well as it can be done in a sick people also. It focuses on improving the knowledge of the disease. So how disease pathology is, how the disease progression is happening. It will be giving inputs in all these characters also. If you want to, it is not only about the drugs. if you want to like develop a diagnostic method, a diagnostic test for a certain illness, then again you have to test that in humans. So the diagnostic methods, the vaccines, the drug, the devices, everything. So if any new technology it is coming and if it is tested in humans, then it will be a clinical research. And new treatment or medical devices, everything it is in which is involving the patient care. So it is very framed and respects a precise study protocol and is only realized under certain conditions. So every drug, the device, the tool, the technology which you are seeing now is once tested in the volunteers who took part in clinical research studies. So again, more in-depth information about the clinical research. So what it is involved? So in that, the disease mechanism is involved. So we will be learning about the etiopathogenesis of a disease, and it is bi-directional integrative research. That means from the lab to the clinical, how the translation is happening. So it is a bi-directional way to understand. And then it gives clinical knowledge, the detection, the diagnosis, and also the natural history of the disease. It will be involving the therapeutic interventions as well. So it can be like biologics or the devices or the instruments, as I have already explained. Then it will also help us in the prevention. Sometimes it will be the development of the vaccine, the testing of the vaccine, or any other preventive measures that can also be tested. And it can also be included as a clinical research. Then behavioral research as well. So how like internal stimuli or an external stimuli will be changing the behavior, how a patient is interacting with the environment, the society, everything can also be learned. So the intervention will be different and what the outcomes will be getting, that will be differently assessed. Then health services research, which is including the outcomes, the cost effective measure. So if you have a same drugs and all, which is more cost effective. So if you want to test in humans and how their satisfaction, everything. So many like multiple dimensions are there for this clinical research. And epidemiological, normal epidemiological study design can also be used. Sometimes it will be for community based, and maybe sometimes it is only the awareness of some sort of diseases and how it is impacting the quality of the life of the patient. That can also be studied. Then now we will focus on history of clinical research. Now we understood what is a clinical research, and now we will look into the history. So in 562 BC, we started with the clinical trials. That is an interesting fact. So now it is very much commonly like many people are doing for the study of new drugs, devices and all. But it started long way before 562 BC. So who conducted this clinical trial? So clinical trial should be conducted by an investigator, their co-investigator, there will be sponsors. There are many professionals involved in the clinical research. But in 562 BC, the king have started this clinical trial. So it is not that well documented, but it is said that there was a king, Nebuchadnezzar, so who was ruling the Babylon. So they have conducted a clinical research where they told their society, the people, to have only meat and wine. So there were some people to object that there are vegetarians, they will object always, like meat and wine will be there, then there is objections from the vegetarians. So the king told the vegetarians to take legumes and water instead. So some one group of people are taking meat and wine, and the other group of people are taking legumes and wines for 10 days. So after the 10 days, the king understood that whoever taken the legumes and the water have more physically fit than the patient, people who have taken with the meat and the wine. So that was the first uncontrolled human experiment done in our history. Okay. And then in 1025 AD, Avicenna's Canon of Medicine, in that dictionary also there are some rules for the CT. So it is regarding to the use of humans, the reproducibility of the results, the time span of the activity and all. So these are the preliminary documentations for the clinical trials or clinical research which is conducted. But the complete systematic review and systematic methodology of the clinical research is in the late 90s. Then in 1537 also, there was an interesting clinical trial conducted, which is accidental by a surgeon. So it was in the wounded soldiers. So the wounded soldiers will have always that boiling oil pour in their wounds. So it will cause fever and severe pain also, even after few days. So like boiled oil, it is very much extreme measure, but there were no other options to treat the soldiers. So when they were out of the oil, this boiled oil, then what they have taken, egg yolks, they have taken the oil of roses and turpentine they have used. Okay, so overnight only fast results they have received. So those who have taken the egg yolks, that the new oil prepared by the surgeon, they didn't had fever, as well as the pain was less comparative to the boiled oil used for the remaining soldiers. So this is an accidental content of clinical trial. Then in 1747, this is the major cornerstone for the clinical research. In 1747, James Lind had conducted a proper clinical trial, but it is not the design, it is not systematical, but still it was a better clinical trial. So it was a scurvy trial with 12 patients involved. So for in 12 patients, they were having a same diet and same conditions and all. So in that 12 patients, for two patients, they have different treatments were given. So in the first two patients, they were given with a quart of cider, and for next patient, it was like vinegar. Next two patients were given with the vinegar, 2 spoons for TID, and then the next two patients were given with vitriol, 25 drops of vitriol TID, and two patients were given with orange and lemon. Two patients have taken the electory surgery given by a different surgeon. So 12 patients, two in a group, have taken different treatment modalities. But after 6 days, the patient who had taken 2 oranges and 1 lemon has a faster recovery. And the second one was for the patient who had taken the cider. So this was then another clinical trial which is conducted and had a proper successful results also. So James Lind had documented this also later in their articles. Then in 18,000, the placebo, the introduction of the word placebo was there, and the placebo related one study was also conducted in rheumatism patients. So there was almost 12 to 24 rheumatism patients. So they were given with the like established remedy plus an herbal remedy. So the placebo, or else in that time there was another word, dummy remedy, that was also being used. So 15,000 placebo word was introduced, and in 1943 first double blind control trial was done. So it was done by the Medical Research Council of the UK, and it was done in the penicillinum patulinum for common cold. But it was not that successful. The results were disappointing, but it was still, the methodology was double blind control trial, where the physician as well as the patient didn't know about the treatment, who is receiving the control and who is receiving the intervention. That was not informed to the patient as well as the physician. And in 1946, first randomized curative trial. It was the streptomycin trial for the pulmonary TB. So that was also like blinded. The nurse was randomly allocating the patients into two. So who is receiving the control care and who is receiving the the intervention care was not given, like not informed to the patient, as well as the physician also didn't know. So in that bed rest and bed rest plus streptomycin was given for the intervention. So that was a successful clinical research we have conducted in 1946. Coming to the Indian scenario. So how the clinical research evolved in India. So from I think from 1911, the first meeting of IRFA happened. So it is the Indian Research Fund Association, IRFA, and they have conducted the first meeting related to the clinical research in 1911. In 1912, the second meeting was held. And from 1918 onwards, they are actively involved in the clinical research. They have done in beriberi, Kala Azar, malaria related. So many clinical trials have been conducted in India in the span of 1918 to 2000. And in 1949, the IRFA renamed to ICMR that we are knowing now. So ICMR is Indian Council of Medical Research. So this is the brief history of clinical research. Moving on to next topic, that is ethical advances in clinical research. So if humans are involved, there are ethical requirements. That is the major importance in clinical research. We are utilizing human. If it is in animals, there are ethical related rules for the animal research, clinical studies, as well as for clinical research, because we are doing this intervention on lives. So it is started in Nuremberg Code. So in Nuremberg Code, only like 10 major points were there. So it is also having a set of ethical research principles for humans. So how you can involve the humans, what we have to do, like maintain confidentiality. So related to that, 10 points were in the Nuremberg Code. And then we are having Declaration of Helsinki also. So ethical principles regarding human experimentation developed in 1964 for the medical community by the World Medical Association. So in that Declaration of Helsinki, it can override any national or local law. So if the Declaration of Helsinki, like whatever the clinical research is doing, it is having a higher standard and safe, then it can override the laws also. So that's what Declaration of Helsinki primarily speaking, and then they will have the other ethical principles also. Then we are having another major thing. It is Belmont Report. So report of the National Commission for the Protection of Human Subjects of Biomedical and Behavioral Research. So it is having three core principles. They are the respect for the persons, the beneficence, and the justice. So respect for the person means whoever involved in the clinical research, we have to show the respect, the proper treatment, and the compensation, the confidentiality, their approval, their consent for the study, everything should be there. And beneficence, the benefit and risk assessment has to be taken into consideration, and justice has to be given to the research, the study participants, and all that. So the area of application of this Belmont report is in informed consent, and then assessment of risk and benefits, and selection of human subject. How you have to select, what is the inclusion criteria, what is the exclusion criteria. So vulnerable population is there, means how we have to include them, or like in what criteria only like we can include them, that and all will be explained. And the next major, it is worldwide, like widely used one, it is the International Council for Harmonization of Technical Requirements for Pharmaceutical for Human Use. That is the major principles we are utilizing, and it is having a QSEM parts. It is divided into 4. So quality, safety, efficacy, and multidisciplinary. So in quality also, Q1 to Q14 points are there. It is all related to the quality of the like what we'll tell product manufacturing aspects. It is coming majorly, and in Q1 and Q14 also there are many subsets also for the same. So in Q1 it can come the stability related studies, the analytical validations and all. So quality related matters. And in safety also, again S1 to S13 points are there. It is also having subsets also. It is mostly based on the toxicology, immunotoxicology, photo safety, and then what we tell toxicological toxicities of the doses, then genotoxicity. So that and all will come in the safety parameters. It is also having S1 to S13 guidelines, and efficacy. Efficacy is the major one which is used in the clinical research aspect. So E1 to E22 guidelines are in the efficacy, and then in the E6 we are having good clinical practices. So that ICH GCP guidelines are very like widely used. So it is having all the clinical trial related things in the efficacy parameter, and we are having multidisciplinary also, that is M1 to M15 guidelines are there. Then it is having MDRA terminologies, then non-clinical safety studies, what are they, then gene therapy. So like the multidisciplinary major guidelines are can be seen in the M1 to M15 guidelines. So it is like freely available. You can go for the ICH site and then you can find out this, and efficacy E6 you can majorly read as a basic parameter. So which is explaining the good clinical practices as like in quality, good manufacturing practices will be explained in efficacy E6 guideline. Good clinical practices are guidelines are explained. Then we will look on to the types of clinical research. What are the different types of clinical research? When you are doing a research, there are epidemiological designs, there are study designs to conduct epidemiological, economical, studies and all. But in clinical research, it is broadly divided into 2. They are observational studies as well as interventional studies. So observational studies means you have an exposure and you have an outcome. So you are observing the patient who are having the exposure and what outcome they are obtaining. So what is the association or correlation between this exposure as well as the outcome that is being studied in the observational studies, and it can identify potential risk factors also, any like long-term effects, or else it is a time bomb. So we can know the long-term effects, ADRs, prognostic indicators, and what is the disease pattern in different population. So all those things can be studied by observational studies. The examples are cohort studies, case control. So it is also involving humans. So it is also a part of clinical research. Then we are having interventional studies, where the clinical research focuses more. In observational studies, the harm is very, very, very minimal because you are not intervening in a patient's life or else the treatment. So you are just observing the patient or the participant or the study subjects. But in the case of interventional studies, we are providing an intervention. We are providing a variable method or alternative method, and we are intervening in the treatment, or else we are using a new treatment, new technology to understand its effect. So in interventional studies, majorly like clinical trials, it is coming. So the major part of the clinical research, we can explain through the clinical trials only. So these studies, whatever the interventional studies we are conducting, are instrumental in evaluating the efficacy of new treatments. What is the efficacy of safety of new interventions? What we are planning, which is already established, but it is not in use of treatment in a specific disease. And what are the new preventive measures? So everything we can, the new it is there that we can learn through these interventional studies. So the like typical major example for interventional studies are randomized control trials. So next we look into the professionals involved. So if you are conducting a clinical research, basically there will be many people involved in that research. So I have taken an example for the clinical trials, like basically like many people are involved in the clinical research. So there are regulators, then there are sponsors and contract research organizations. So contract research organization is a major term which you have to remember. So they are fully independent and they can conduct a complete clinical trials. So they will be taking up some studies. So like clinical trials, if anyone is planning to do the clinical trials, you can give it to the like CRO completely, and they will be responsible for this. And sponsors will be giving this giving these clinical trials to the CROs, and they will be sponsoring the whole study. And under the CRO, there will be medical director, the whole project manager, how it has to go, like protocol development, all the other documentation, everything. The project manager is having huge responsibility. And the next major one is CRA, clinical research associate. So they will be explaining the how a study has to be conducted. They will be communicating with the investigators, sub-investigators and all, how to plan the study and monitor the clinical sites where the study is being conducted, and continuous monitoring and follow-up of the study, the documentation, whether it is happening properly, everything it is overlooked by the CR. Then there will be a regulatory personnel to do the regulatory related documentation, to overlook the regulatory aspects of the clinical trial as well. And then investigators or sub-investigators, they will be the person who are in the clinical trial, who is taking the informed consent, who is allocating the participants, giving the treatment, everything it is done by the investigators, and they will be presenting the study in front of the Institute Review Board. So ethical approval, it is very much necessary to conduct this clinical study. So investigators will be getting that approval. And then CRA again being the bridge between the subject and investigators also, they are also helping in that communications. So for an explanation, whatever I explained in the previous slide, the same thing it is coming, but more example, I can give you now the sponsors. Sponsors include the private and public center. They are funding organization. So the major largest clinical research sponsor is NIH, National Institutes of Health. So they are the major clinical research sponsor. Then we are having Department of Defense, Department of Veterans Affairs. They are funding certain clinical trials also. Then we are having research organization who are conducting this kind of studies. So in research organization, we are having academic health centers. So if any medical colleges are, they can be there. Private research institutes can also be there. In India, there are many. Then survey research organizations are there. Federal government intramural research programs are there, and contract research organizations also. These are different research organizations which are carrying out these clinical trials. Then we are having investigators. So as I explained, scientists performing clinical research from varied disciplines with a range of academic qualification, they will be in the clinical side. Okay, so they can be having academic qualifications of MD, like PhD, then PharmD. Everyone is eligible for the being an investigator for the clinical trials. Then oversight entities who is looking after this sites and the clinical trials. There are IRBs, then FDA, Department of Health and Human Services, Veteran Affairs, National Committee for Quality Assurance. Everyone will be overlooking into this clinical trial, whatever it is being conducted. Then contract research organization. So they are kind of outsourced research services. They are providing outsourced research services to any pharmaceutical companies like biotech companies or medical device related companies. So these manufacturing companies has to do some sort of clinical research to test their product. So the contract research organization can take up the clinical trials and look after all the documentation, regulatory, and the trial related requirements. So some examples for this contract research organization as in India, like in majorly in India also the branches are the IQVIA, Paraxel, Covance, and all can be the examples of contract research organization. Then we are having clinical research associate also. So person delegated by the sponsor. They are working for the contract research organization. They follow and control the progress of the clinical study at the experimental centers. Okay. Then there are participants. They are the volunteers, healthy volunteers or sick volunteers will be there. So they will be participating in the study. They might be the patients also and population at large or healthy volunteers. Then the stakeholders or consumers. So after all these personnel involved, to whom we are reaching out, for whom we are conducting all these things. So there are stakeholders, again, that is the society, the major one, and then the consumers also. Some health insurers needed this, manager care organizations will be there, healthcare system requires this clinical trial related informations, then voluntary health agencies, patient advocacy groups. So there are many consumers for these clinical trials results. So now we go to know what is clinical research, how it evolved, what are the persons involved in this, what are the types. So before coming into the clinical research, what is the drug discovery and development process? How this new entity is formed, that we will like shortly, like in a brief way, we will see that. So at first, the starting of this like new entity, new molecule, it is from the target identification. So first we will see in a disease which genes are overexpressed, which protein is causing it, or else which enzyme is causing this disease. So identifying the target is the first one, and we will be scanning, like now there are huge chemical databases and all. So we can screen the chemical databases to get the lead molecules, whoever targeting the identified target. So first we have to identify which target we have to act on. So there are some examples of this chemical databases, that Sanger Hall Genome CRISPR library is there, and Duolink PLA, they are major databases, examples you can say. And then once the target is low, then lead discovery is there. We have gone through, we have scanned a lot of chemical databases and all, and then we will see which chemical interact with the target which we have identified and which is having the positive pharmacological effect. So this lead discovery can involve many methods, that high throughput screening, hit to lead method, virtual screening, and natural product screening, and all other different methods for the lead discovery. There will be like thousands of molecules and all. From that, we have to filter and come into very, very few. Always it will, after many laboratory related investigations, it will be very, very, very few molecule which is getting into the last stage. Then once we have identified the lead, like what molecule is better, like more effective, then we will be optimizing that lead molecules. So that lead optimization is done for the like pharmacological activity, selectivity, the safety, and structure. Everything will be modified in this lead optimization. So we can give a more effective way for the new entity. Then it will include in this lead optimization, may the subject area medicinal chemistry, it is structural biology, the structural changes, whatever has to be modified, and the pharmacology, pharmacodynamics and kinetics has to be seen. So once the target is identified, we will discover the lead molecule. After the lead molecule is being discovered and lead, that will be optimized and it will be given for the animal studies. That is preclinical testing. So this will take over like many years, 10 years, like that much time also it can take. So then preclinical testing will be done in the animals. So it will be giving the like safety, efficacy of the drug in the animals. So we can learn more about the pharmacodynamics, pharmacokinetics, the toxicology related aspects of the drug molecule. Before giving it to the humans, we will test in the animals. Like sometimes it will be starting from rat, mice, guinea pig and all. It can larger order animals also being tested like monkey, dogs and all. So then we will get this data. Now one more like new way it is there. We will extrapolate this animal data to the human data using data analytics and other major like new methods, technologies and all. So we can, before giving it to the humans, we can identify how it can react or it might react in the humans. So after these 4 steps comes our clinical research process. Whatever after these 4 steps, it is clinical research process. So in clinical research, majorly we will be discussing about the clinical trials, because once the preclinical testing is done, then it reaches to the human phase. So human phase means it is clinical trials. After clinical trials, it will go to the market and all. Then post-marketing surveillance is actually the phase 4 of the clinical trials. So after that comes the observational studies. So we will see what is clinical trials and how in each phase, what it is happening. So clinical trials, the phase, first one, it is phase 0. So it is micro dosing in humans. Only a micro dose will be given to the humans, and it is in very, very small number of participants, only 10 to 15, because we do not know. Like even though we have conducted preclinical testing or extrapolation of data, but still it is kind of speculation, like it might, it will, it react like this. So that kind of like doubts we'll be having. So we will be giving only for a small number of participants, that is 10 to 15 participants will be giving that. And we will gather the preliminary pharmacokinetics and dynamics in humans, how the drug it is acting in humans and how the human body is acting to the drugs, that we will be checking. The preliminary data will be gathered. We will not know like a major like a population at large. If you're giving this medication, it can react in very, very different methods. But the preliminary data we'll get from this phase 0. So we will not get any data of safety and efficacy. We will understand how it is acting and whether it is safe or not, but we will not get a significant or scientifically significant data for safety and efficacy. So then we will be conducting, once it is success, then we will be going for phase 1. So that is the first stage of research. So we will taking a little larger number of participants, that is up to 20 to 100. So we will be learning the safety of the drug. So entity like the drug or the vaccine or the device, whatever it is there, whether it is safe to use. Then toxicity, pharmacokinetics we will be checking, and dynamics in humans we will be looking after. Then the dose ranging also, in what dose we have to, what is the therapeutic range and what is the toxicity range, that and all preliminary data will be obtained from the phase 1 clinical trials. Then you are having phase 2 clinical trial. It can also be called as therapeutic exploratory trial. So approximately like we are taking 20 to 300, from 10 to 100, we are taking the number of subjects, and so we will be checking here efficacy and dose optimization. Phase 0, whether it is safe to use and how the humans are reacting to it. In phase 1, we will be checking the safety and the toxicokinetics part and all. And in phase 2, we are checking the efficacy, how effective it is, how potent it is. That and all will be checked in phase 2. So dose optimization, also the proper therapeutic range and all has to be checked. So that will be done in this phase 2 and all. And patients with targeted disease are our subjects. So whoever having the disease, whoever having the requirement, for those patients, we are giving this in phase 2. And then in phase 3, it is therapeutic confirmatory trial. So after all these things, we are confirming that the new molecule or else a new drug, the new medical device, the technology, it is safe and effective to use. So in that 100 to 1000, like 300 to 3000, or else you can like broadly tell 100 to 1000 participants will be there. And we will get a sufficient scientifically significant data of safety, efficacy, and all the things related to the new innovation. So it will also assess the long-term safety. Then we will be analyzing the risk and benefit associated with the new innovative method. Then once the phase 3 it is completed, it will be marketed. Once the regulatory approvals are obtained and everything it is satisfactory, then it will go to the phase 4 clinical trial. Phase 4 clinical trial can also be called as post-marketing surveillance. So in post-marketing surveillance, we will be surveilling the safety of the new method. So it can, like a major one, it is pharmacovigilance, we can tell. So in that, if a new drug it is getting marketed, then what we'll be doing, we will be checking the long-term effects. If the drug is taken for 5 years, 10 years, 15 years, what is the effect? That can be studied only in the populations. So the larger population. So the long-term effects can be seen, and the adverse drug reaction in phase, like in clinical trial phases and all, we have received like a adverse drug reactions, many reports we will receive. But apart from that, when it is giving to the different population, different individuals and all, how like new drugs can, like adverse drug reactions can be identified, that and all it is happening in the phase 4. The major problem, like the major challenge of the post-marketing surveillance is underreporting. Because in the phase 0 to phase 3, all the patient participants are with us. There are project managers, investigators, CRA, CEO, CRO, then oversight entities. Like there are many people to monitor the clinical trial. And all the, like in all the phases, there is a specified number of participants also, which we can trace. So there is not much problem. But when it is coming to the post-marketing surveillance, when it is given to the whole public, then we are having problem with tracking. So if any adverse drug reactions are already happened, but it is not reported to the, like in India, it is PGPI. So it is not reported to the PGPI. Or else the, like because of underreporting, the frequency of adverse drug reaction can also be miscalculated. So there are many like problems when it is coming to the larger population in the like tracking of this. Then action of, like we can check when we are having it with milk, when we are having with any acidic food, what it is happening. So what is the food drug interaction, drug interaction? So what are the other interactions when it is coming to the market, that can be also studied. So real life situations, it is like numerous. So in all the real life situations, we can study about the new method or new intervention, new devices, technologies, etc. Next, we will look on to the what are the major points, what is the like regulatory, as well as how clinical trials are going on, and what are the regulatory requirements in each step. So once the preclinical data it is there, whatever the laboratory findings and like animal testing details, all the things will be submitted in the IND filling. Investigational new drug applications are there. So IND filing has to be there. And as soon as the IND filing it is there, FDA will be like responding to it, and maybe like within 30 days also, after that, after 30 days after reviewing also, you will get the response. So once the data of the laboratory findings has been submitted, the complete documentation has to be there, that should be filed and submitted. And once the IND approval it is received, then we can go for the phase 1. Before going phase 1, we need to get the ethical approval of the sites. The IRB has to be completed. That is the point 1. After phase 1 also, phase 2 also, phase 3 also, we have to go for this regulatory requirements. So whatever the data received from the phase 1 has to be submitted to the regulatory bodies, then only you can proceed for the next step. Again, in the phase 2 also, you have to submit the documentation. So clinical data management is a very huge importance in clinical research, and career opportunities in clinical data management is also very high. So in this phase 1, phase 2 and all, like the sponsors are carrying out this clinical trials. They are the major person who are going for these clinical trials. If the results are not satisfactory, and if it is not getting a desired benefit, desired outcome and all, the sponsors can decide like whether to go further or not. So after phase 1 information, phase 2 information also they can. First, the go, no-go decision will be taken in the preclinical data. After preclinical data, it is not satisfactory, it is not benefit, they can stop their research there itself. And again, after the phase 1 also, if the limited population 10 to 15, it is harmful and if it is not having a successful outcome, then you can also decide. The sponsors will be deciding whether to go further or not. So then till phase 3 also we are going for this. Then after the phase 3 it is done, then we are filling an application for NDA, new drug application form. So NDA will be filed and we will be waiting for the NDA approval. After the NDA approval, then marketing permission has to be required. So again, the regulatory bodies everywhere, the IND filling, NDA filling, everything, it is of regulatory requirement. So that during the marketing permission also, you need the regulatory approval. Then it will go for the phase 4. That is the drug, it is marketed. So in all these regulatory requirements, you might get many suggestions also. Maybe it is related to the labeling of the things, how in label it should be mentioned, the scheduling, everything. So many suggestions can be obtained during this time period. So this is the whole process of clinical research. So now we will see what are the process for the clinical research. So this is like a clinical trial, we can tell. So the complete process of clinical research with regulatory we have seen. Now we will go on to in a clinical side, or else one clinical trial you are doing, what you have to do. So that we will see. So first you have to determine the study objective. If you have a problem statement in your mind, the hypothesis is ready, then you have to determine the study objectives. What are the objectives of the study? Then you have to prepare the whole protocol where methodology is the very major part. So background study, the literature search, everything will be there in the protocol, plus the detailed methodology, what materials you are using, what study design you are using, how many sites are there, like what is the intervention, if any, all the intervention has to be detailed, and what is the study procedure, and whether what are the ethical considerations for that, what is the workflow, and if any dose it is there, all the calculations related to the dose, the use, the benefit, all the data about that specific drug, or the drug monograph kind of thing also has to be done. So in the methodology, each and every detail about the product you are using, the method you are going to follow should be specified properly, and the statistical analysis that you are going to do. Then you will be, once the protocol, the complete study design, the study procedure is set, then you will go for the site recruitment and the investigator. So site will be one hospital and investigator can be from the same hospital itself. Okay. And then prepare the case report forms and all, whatever you need to do, the data collection and all. Then you have to prepare the informed consent form. Only after obtaining the informed consent, you can go for the enroll the participants into the study. So in the informed consent also, the process of getting informed consent is also very, very, very important. So that means it has to be in different languages, especially like where the site is according to that, their local languages. The informed consent should be translated to their local languages, and all the informed consent procedure that should be recorded also, the audio, video. So before recording the audio and video, you have to take the consent from the patient to record that also. But it is very of importance. So each and every step which is explained here is also having very much detail in itself. And then after apply for IRB approval at each site. So once the site is an investigator, it is ready. And when you are before going to start the study, you have to apply for the IRB of that specific site. If it is a multi-center, all the center related IRB has to be received. Then investigator agreement at each side, whoever the investigators are there. So that has to be done. Then apply for ID is also there. It is that device related. So investigational device exemption that also need to be required if in case you are going for the device related intervention. Then you have to then in the clinical trials method, then investigator meetings, the regular follow-up and monitoring and investigator meetings will be there. Then the conduct of the study will be simultaneously going on. Then collection and documentation of data. It is a very huge area. And if you are documenting in each date also you have to mention, and you cannot one major point but I want to share is once you have documented anything, you cannot strike through it. You cannot erase and write a new data to that. You just one normal strike you can put. So what you have written previously and what mistake happened in that and what you are writing, everything should be clearly evident. So the clinical data management is also a major part. So documentation and management. Then analyzing of this data will be there, and the report writing and presentation will be there. So this is the complete clinical trial or a randomized control trial, whatever whatever trial you are going to do, you have to go through all this process. Then the major three documentation and regulatory related requirement, that is IND application. So after preclinical studies and all, we will be applying for the investigational new drug application for each and every clinical trial. Before conducting the trial, you have to get this IND approval. Then we will be going for new drug application. Once the clinical trials have completed and it is having satisfactory result and it is having a very minimal risk and benefit is more than the risk, then you can go for the new drug application. Then the drug sponsors formally propose that the FDA approve a new pharmaceutical for sale and its marketing. So after clinical trial, you are going for the new drug application. Then the data of animal studies, human clinical trials, everything has to be submitted during the new drug application process. Then there is one more thing, that is abbreviated new drug application. So IND investigational, ND new drug application, and abbreviated means what? That means already under a brand name, the drug has been undergone the clinical trials, the preclinical testing and everything, and it is being marketed. So if you have to produce a generic drug or a bioequivalent drug, then you can go for the abbreviated new drug application. So it is a very good thing, because when already a manufacturer of a brand named drug, so if they have gone for this clinical trials and all the process has been completed, and when we are going for abbreviated new drug application, it is majorly it is done for the generic drugs. So cost-effective parameter is coming into the picture also. So it is having many advantages like that. So in ANDA, there is no requirement for the preclinical and clinical data. There is no need to submit all these data. So the other people have done this research, that data there is not necessary to submit, but bioequivalent studies are most important. So we have to prove that whatever we have taken the new entity, so that is bioequivalent to a different active ingredient, different API, which is already been marketed. So that bioequivalent studies has to be completed. So in that also you can find the major what are the already marketed drugs. There is RLD document, that is reference listed drugs. So that document you can go through and you can understand it's what it is already marketed, and then go for the bioequivalent studies. So coming to a little bit of conclusion, we will see what is the importance of CR. So we have explained what is CR, what is the process and how we have to conduct it. But what is important? So it is having major advantages. So it is giving a major insight into the treatment decision and enhance the quality of care. Already pre-existing treatment, everything will be there. So if a new method it is coming, so it will be more effective. Then only it will be marketed and it will be taken into the treatment care. So like a good decision, treatment decisions can be made based on the data available, and evidence-based practice can be implemented. So like new drugs can be like whatever the new drugs can vaccines, drugs, devices can be evaluated, and you will get regulatory approval for new medicine if you're conducting the clinic. Only if you're conducting clinical research, you are getting the regulatory approval. So then like evidence-based guidelines can be done, and healthcare providers to deliver personalized and effective therapies also according to the individual genetic makeup also. Like if new methodologies are there, you can provide personalized therapies also, and development of innovative medical technology. So advancements in the medical field has to happen through clinical research only. Because like whatever new things it is happening in medical field, let it be the technology, the devices, the drugs, everything should undergo clinical research. So clinical research is the basis of advancements in the medical field. And you can implement new public health policies, resource allocations, and all based on the clinical research data. Then, what are the challenges and future of clinical research? So, once a clinical research is having many like advantages, they might be facing certain challenges also. So, what are the challenges faced by the clinical research, and what will be the future of clinical research? So, the challenges are complexity, the complexity of study and the cost of clinical trials. Like if many like personnels are involved and many documentation has to be taken, compensation if any harm it is there. So like then many things are there, and it is very time consuming also. So the cost of clinical trials are very huge. So if you're not having that budget, then you cannot go for the clinical trials. Even if you are having a new entity, still you are not able to get the regulatory approval or get it marketed, because maybe the budget, the cost, and the complexity of these procedures. Then recruitment and retention of diverse study population. So public awareness is limited at this time. So like recruitment of the patient into the research, that is a major issue, and the retention also. If you have to follow up them for a longer term, then retention rate should also be high. So dropout rates should not be high when you're going for a clinical trial. So if dropout rates are high, then the validity of the study is also reduced. So recruitment and retention is another parameter. Again, recruitment is happening in healthy volunteers based on the monetary benefits also, so that many people are getting ready. So these can be changed when the public awareness is there. Then need to accelerate the translation of research findings into clinical practice. So whatever the laboratory findings are there, that has to be translated to the clinical practice always. So many of the times that are underdiscovered, and it is not like translated to the clinical practice. It is time consuming, of course. It is time consuming also. Data integrity and management, that should be of higher higher standard. So the management of the data, the integrity of the data collected should be there. Then public awareness should be enhanced. That is the major challenge of clinical research. The people are not knowing what is investigational drug, what is a new drug, or like maybe like in a simpler format, we can explain them so that the public will be aware about what is happening in the medical field and how it is benefiting them. Then intellectual property and transparency issues about the data and intellectual properties. So there conflict of interest within the authors. So like that, many other major things are the challenges of the clinical research. Then what are the future perspectives of clinical research? So there are developments in the data analytics. So in the data analytics, so extrapolation will be majorly easy. Then artificial intelligence is also taking place now. Like it is very higher rate in growing. So artificial intelligence can also put into use for the clinical research. So that will be the next future of the clinical research. In many places, it can be like we can do the artificial intelligence related technologies. Then the precision medicine targeted like gene therapies and precision medicines is also a future of clinical research. Major clinical research will be happening on the precision medicine in the near future. Then global collaboration, so multi-center studies are there, but it is now also limited. The number of like global collaborative projects are less. So if in India we are doing a project, the multi-center project, all the centers will be in India itself. So that pattern is seen now very much. But in future, global collaboration can be enhanced, felicitated, so that you can learn about the clinical research in the diverse populations. So in the different race, ethnicity, so everything can come into one umbrella when the global collaboration is there. Then digitalized documentation, the error in documentation and safekeeping, safeguarding of the documentation can be more easier when it is digitalized. Otherwise, missing reports can be there, the entry or misinterpretations, or else if one person is telling and the other person is writing, that should not happen when the clinical documentation is there. And if it is happening, then it can lead to many errors. So man-made errors can be reduced in documentation when it is digitalized. So these are the challenges and future of the clinical research. So thank you so much for listening. So I hope you have got the some knowledge or some insight into what actually the clinical research, what the process is, the types, the importance, and what are the challenges and futures. So if you are having any question, you can reach us at the email IDs and all the social media handles it is given. So if you are having any kind of doubts related to this topic, you can always reach us to, and we are always happy to help you all. Thank you.
Thank you so much, Dr. Harita, for enriching and engaging session. Your insight have truly added a great value to our understanding of clinical research. Being a clinical pharmacy practitioner, I do have a few questions on behalf of our clinical pharmacy or budding clinical pharmacist. The query is nothing but usually PharmDs used to think that the clinical pharmacy or clinical pharmacy profession is only for on behalf of academics, or we have to practice as a clinical pharmacist, because always we restrict ourselves from being a just a clinical research just only to the clinical pharmacy or academics. What do you think? Like, what are the skills or essential skills we need to enter into clinical research, or is it possible to join soon after the PharmD into clinical research?
Okay, that was a good question. So my viewpoint in this is basically when we are discussing about the PharmD graduates. So PharmD graduates are the most suitable person for clinical research. That's what I feel. PharmDs has to explore these kind of job roles, because I have told the academic qualifications for the clinical research personnel. So investigators can be from MBBS background, MD people, DM people and all, but they are more busy in focusing on the patient care. So the practicing of their things, and then they have to focus on how you have to give the best quality of life for the patients. So whatever the existing guidelines are there, the existing treatments are there, that they are providing, they are having many patients and they have to save many lives. So the doctors are usually busy in doing that part. And when it is coming to the B Pharm and all, so they are according to their domain in the M Pharm, Master of Pharmacy, they are busy in manufacturing field, or else ensuring the quality control, or preclinical. So whatever the drug discovery or development process, the first 4 steps I have explained. So the major roles of M-Pharms are happening in there. But when you're coming to the clinical pharmacy department or pharmacy practice department, where M-Pharm, pharmacy practice and PharmDs are there, they have the major knowledge about the pharmacology, pharmacodynamics, and the pharmacokinetics, biopharmaceutics, like that, many subject information they are having. So they are the suitable for person to conduct these clinical trials. The ethical requirements also they are in non the study designs. So about the research, vast knowledge is for the PharmD people. So they can be investigator for all these things. They can take up the role of CRA also, project managers also. So they can handle these kind of clinical research very beautifully. So I hope I have answered your question. Like, am I clear, or any other queries you're having?
Yeah. Yes, clear. And one more query which I have is nothing but since we are from a pharmacy background, our syllabus is not that updated with the clinical research or anything, like just biostatistics and research methodology. So whenever we go through the syllabus of means clinical research program, everything will be a bit different, and the exposure, we doesn't have much exposure. We have exposure only to a limited time, that too in academic research. To get an exposure to the clinical research, only few are taking internship. Like other than the syllabus, they'll go outside some MNCs, they'll do internship and they'll get exposure and they'll get into clinical research. So other than that, how a student, a budding pharmacist or clinical pharmacist get an exposure to clinical research? Like, is there any special programs or online platform you would like to suggest for early professional, career professionals or beginners?
Okay, that's a great question. Actually, like the syllabus for PharmD in clinical research, it is like a little bit limited, and it is majorly focusing on the clinical trials, the regulatory requirements as such. So there are more into this clinical research. So in like 120 hour of clinical research syllabus in the PharmD, which is designed in the 2008 year, and it is not that much revised. So in that 120 hours, we will not receive the adequate skills or knowledge related to the clinical research, conducting of a clinical research. So it is always important to learn beyond the
designed in the
syllabus.
2008 year, and it is not that much revised. So in that
So
120 hours, we
what
will
we
not
have to
receive
do
the
is like
adequate skills
there are many
of knowledge
certification programs.
related to
I
the
cannot like suggest certain certification programs or courses related to the clinical research, but surely I can tell what to look into these kind of certification courses. So always when you're going for a like a course, it should be more than 30 hours. At least at the minimum 30 hours should be completed. Otherwise, we cannot ensure that you have received that a certain skill. So that like 90 hours, 120 hours, so like specific document you have to take. If clinical trial it is there, like in phase 1, phase 2, what is happening in depth, you have to learn for a like multiple hours, do assignments and projects related to the same. And then like there are many free like webinars, seminars going on in the name of the clinical research, as well as in the name of certain subfields in the clinical research. But I'm not sure, like adequate knowledge will not be received. A basic knowledge can be received, basic idea about the topic can be received, but you can never achieve that skill through that 1 hour, 2 hour, or 1 day workshop. So for that, you have to practice, as well as you have to know the all aspects. So clinical data management is the major thing. The process of like documentation of the data, the management handling of the data, and all are a major important part. That is one major skill set you can receive through some additional courses. Then the process of clinical, how to develop a protocol, what should be the content. I can only briefly explain in this 1 hour session. I can only briefly explain what is in the clinical protocol, but the each section is again having like subsections, and what has to be included in a protocol, that is again a major vast area to learn. So you have to go in depth of each and every step in clinical research and be expertise in the steps. So that requires a lot of time and practice. And then you can like ICN GCP guidelines, you have to go through all these guidelines knowledge also is very, very important. Not only ICN GCP, ICH related guidelines in that efficacy part, it is majorly for the clinical research. So that kind of guidelines, the Belmont report, the Declaration of Helsinki, so that kind of principles and the regulatory requirements you need to know. So regulatory assets is a very like major area in clinical research. So you can just only focus on regulatory, or else focus on clinical data management, focus on the process of clinical research. So subdivide these sections, and you have to learn keenly into these kind of topics. Then only you can acquire that skill and you can excel in that field. So I think I have explained the answer, like it is sufficient to answer your query. So I think I have explained that.
Yes. Yes, thank
Answer.
Yes, thank you, Dr. Harita. So, audience, if you have any question regarding clinical research or the session, you can reach us at fcpiofficial@gmail.com. So, thank you, Dr. Harita. On behalf of the Federation of Clinical Pharmacists in India and the Clinical Research Forum, we extend our heartfelt thanks and appreciation to you once again for sharing your expertise with us. Thank you, Dr. Harita. So, thank you,
Thank you.
the Federation of Clinical Pharmacists in India and the Clinical Research Forum. We extend our heartfelt thanks and appreciation to you once again for sharing your expertise with us. Thank you, Dr. Harita. So, thank you.
Thank you.
The Federation of Clinical Pharmacists in India and the Clinical Research Forum. We extend our